In a previous video, we discussed the diversity of fermentation pathways in bacteria. We discovered that some of these fermentation pathways not only regenerate NAD+, but can also produce ATP. In anaerobic bacteria, ATP is not only produced by glycolysis and some specific fermentation pathway. In this present video, we will discuss how under anaerobic conditions, bacteria use a process called anaerobic respiration to produce much more ATP. And we'll focus on one type of anaerobic respiration that has a significant impact on our economy and our environment. Before we start, let me remind you some terminology. First, we have cellular respiration. That is a process that is an aerobic process. Glycolysis and citric acid cycle are active. Oxygen is used as the final electron acceptor during oxidative phosphorylation. Then we have fermentation. That is a process that happens in both anaerobic and aerobic organisms, when these organisms operate at low level of oxygen. In this case, fermentation will increase the rate of glycolysis and provide an alternate pathway to regenerate NAD+. Under this condition, oxygen is not required for fermentation. Then you have anaerobic respiration, which occur in anaerobic organisms that grow in the absence of oxygen. Glycolysis and the citric acid cycle will occur, but oxygen will not be the final electron acceptor. The final electron acceptor will be an inorganic molecule. In fact, ATP production by anaerobic respiration is very similar to the production of ATP during oxidative phosphorylation. A gradient of proton will be built, and the energy stored in this gradient will be used to drive ATP synthesis. What are anaerobic bacteria? First, these are bacteria that grow in the absence of oxygen, and we can classify them based on the type of final electron acceptor. For example, this table shows you that denitrifier will use nitrate or nitrite as an electron acceptor. Sulfidinogen will use, for example, sulfate as a final electron acceptor. And methanogen will use carbon dioxide as a final electron acceptor. The final electron acceptor will determine the nature of the final product of anaerobic respiration. If we take, for example, the case of cellular respiration, the final acceptor of electron is oxygen, and the final product is water. With anaerobic organisms, let's take, for example, denitrifier. The final acceptor of electrons is nitrate, and the final product of anaerobic respiration is dinitrogen. We can also see on this table the change of free energy for each of these anaerobic respiration reactions. And we can see that the most exergonic respiration occurs in denitrifiers. What does that mean? So let's take a scenario where all the anaerobic respiration that are listed in this table are present in the same environment, and that all the final electron acceptor are plentiful. Under this condition, denitrifiers will flourish because they will produce a lot of energy through respiration and produce a lot of ATP, more than the other organisms. So they will predominate. They will have a competitive advantage.
Are there always more denitrifiers than other anaerobic bacteria?
The answer is no. And you bring a very good point. thermodynamics give you only a part of the whole picture. Bacterial proliferation is dependent on the availability in the environment of fuel molecules and electron acceptors. And it turns out that in most environments, metal ions are more abundant than nitrate, which means that in all of these environments, the metal reducer will be predominant and not the denitrifiers. However, we will focus on the denitrifiers due to their impact on our environment and our economy. So what is the impact of these denitrifiers? First, we have to define what is the denitrification reaction. This reaction is the process by which nitrate will react with protons and electrons to produce dinitrogen. Microbial denitrification results in a loss of nitrate in farmlands. And nitrate is a critical nutrient for the growth of plants. So if farmers want to maintain a high yield of crop production, they need to spend a lot of money to buy fertilizer that will bring back nitrogen to their field. Now, a substantial part of these nitrate-based fertilizer, as well as phosphate-based fertilizer, that the farmers had to their field, will be washed away and accumulate in rivers and lakes. And you have to remove nitrate from drinkable water. That's the function of many wastewater treatment plants. And some of these plants use denitrifiers to remove nitrate from the water. So why nitrate has to be removed from drinkable water? Well, an excess of nitrate in the water will cause health problems, in particular in babies. If you remember, hemoglobin can get oxidized into what is called methemoglobin, where the iron in its ferric 3+ state. And this hemoglobin cannot bind oxygen. When nitrite enters the red blood cells, nitrite will cause the oxidation of hemoglobin. So if you have an excess of nitrate or nitrite in drinkable water, babies will accumulate methemoglobin more than adults. And the consequence will be deoxygenation of tissues in babies that will lead to a form of what is called the blue baby syndrome, which has dramatic consequences and can be lethal. Now, if nitrates accumulate in bodies of water, then it will stimulate the growth of phytoplankton. Because under normal conditions, what limits the growth of phytoplankton is the fact that nitrate is in limited amount in this water. So if you have a large quantity of nitrate, phytoplankton overgrow and consume oxygen. And the hypoxia in the lower layer of water will cause the death of aerobic organisms, for example, fish. Now, let's try to figure out how nitrate is converted into dinitrogen. Denitrification is a stepwise process composed of 4 sequential reduction reactions. First, nitrate is converted into nitrite. Then nitrite is converted into nitric oxide. Nitric oxide is then converted into nitrous oxide. And finally, nitrous oxide is converted into dinitrogen, which is a gas that can diffuse in the environment. All of these reactions that are reductions will require electrons. And the electrons will be supplied by membrane-bound complexes. What are the enzymes and cofactors that are involved in denitrification? Despite structural differences, both gram-positive and negative bacteria have a cell membrane that separates the cytoplasm from the periplasm. It is in this cell membrane that you find the first electron acceptors. In fact, the first one that interests us is a molecule called coenzyme Q, which is extremely hydrophobic and is embedded in the cell membrane. Coenzyme Q receives electron and protons from different universal electron donors, for example, NADH. Coenzyme Q, when is reduced, is also called ubiquinol. So ubiquinol will be then the molecule that diffuses within the membrane of the cell and transfer its electrons to the first enzyme that catalyzes the first reaction of denitrification. Ubiquinol will interact with the gamma subunit of an enzyme called nitrate reductase. The electrons will be transferred to this unit and will travel through the 3 subunits of the reductase. The protons will be released from coenzyme Q and will end up in the periplasm. So that transport of proton in the periplasm will contribute to establishing the proton gradient across the membrane. When the electron reach the alpha subunit of the reductase, which is on the cytoplasmic side of the bacteria, these electrons will be used to convert nitrate into nitrite. This reaction will also consume 2 protons because a molecule of water is formed. That will further contribute to the proton gradient across the membrane. So now what happened to nitrite? The nitrite molecule can then diffuse from the cytoplasm to the periplasm using first a very simple transport protein. Then a second transport protein will come into play. And this protein will allow the transport of nitrite outside of the bacteria, coupled with the entry of nitrate inside the bacteria. Therefore, this transport protein will regulate and coordinate the import of nitrate and the export of nitrite, and will limit the accumulation of nitrate inside the bacteria. Now, the rest of the denitrification reaction will take place in the periplasm. The first reaction is a conversion of nitrite into nitric oxide, and this reaction is catalyzed by the nitrite reductase. The third reaction is the conversion of nitric oxide into nitrous oxide. So 2 molecules of nitric oxide will be needed to produce 1 molecule of nitrous oxide. This reaction is extremely rapid to prevent the effect of the toxicity of nitric oxide. Finally, the fourth reaction is the conversion of nitrous oxide into dinitrogen. This reaction is catalyzed by the nitrous oxide reductase. Dinitrogen, which is a gas, will then diffuse outside of the bacteria and will accumulate in the environment. All of these last 3 reduction requires electrons. Remember that with the first reaction of denitrification, electrons were provided to the reductase by ubiquinol. But ubiquinol is extremely hydrophobic, and it cannot deliver directly its electrons to the reductase that are found in the periplasm. In fact, ubiquinol will transfer its electrons to a membrane-bound complex called complex 3. The electrons in complex 3 will be transported toward the periplasmic side of complex 3. On that side, in the periplasm, a molecule called cytochrome C can interact with complex 3 and be reduced by the electrons that come from complex 3. Then, cytochrome C in its reduced form will then interact with a different reductase in the periplasm and transfer its electrons. So cytochrome C is in fact an electron shuttle between complex 3 and the reductase. The protons that were also released by the oxidation of ubiquinol end up in the periplasm. Furthermore, the transport of electrons through complex 3 will be coupled with the transport of proton from the cytoplasm to the periplasm. Once again, this transport of proton will contribute to the establishment of a proton gradient across the membrane. While some protons are consumed in the periplasm, there is an overall buildup of a proton gradient across the membrane. This proton gradient, as we will discuss later in this course, will be coupled with the synthesis of ATP. In conclusion, denitrifying bacteria use nitrate as a final electron acceptor. Through a stepwise process, nitrate will be reduced into dinitrogen, and the energy that is needed for ATP synthesis will be produced by the establishment of a proton gradient. This example illustrates the existence of pathways other than glycolysis and fermentation to produce much more ATP in anaerobic bacteria. We will discover later some similarities between the production of ATP through anaerobic respiration and through oxidative phosphorylation. Don't be fooled by this simple example of metabolism in bacteria. In fact, bacteria rely on a wide variety of carbon sources and electron donors to produce ATP, but they also rely on inter- and intraspecies cooperation.